Last year, the Nordic Network of Astrobiology, together with the NASA Astrobiology Institute, organized a summer school in Iceland for 2 weeks, and one of the projects was to test the colonization of different environments by microbes. The data we got at this time was interesting and intriguing, and so some students and young scientists decided to do a follow-up expedition to explore their questions which were involved a little bit further.
We were here to demonstrate some techniques as part of that summer school, and in fact the majority of the people who were here on this campaign are a subset of the people who were at that summer school, and decided along with ourselves that it would be worthwhile to then organise a further expedition to Iceland this summer to start developing further the application of these techniques to this particular location.
It's a group of about 45 different graduate students and postdocs studying astrobiology, and we had a really great time here, kind of both doing lectures and also field sampling.
I really enjoyed it. I enjoyed meeting astrobiologists from around the world, students from Europe, across Europe and the Americas.
The summer school last year was really a lot of fun. Got to meet a lot of people from different parts of the world, from different backgrounds, a lot of different expertise. Last year we did a pretty good job of introducing ourselves to a bunch of useful field techniques, and I wanted to learn more about those and get some science done.
Hello, my name is Wolf. I'm associate professor at Stockholm University in Sweden, and I'm also coordinator of the Nordic Network of Astrobiology and director of the Stockholm University Astrobiology Center, which exists since 2011. I was more or less functioning as a kind of local coordinator and local scout for the different logistics also, since I am involved in Nordic astrobiology research and thus have a certain experience with Iceland and with also the Icelandic bureaucracy and the red tape you have to do for such expeditions, which can be quite substantial. For example, for our expedition, we needed 5 different permits for each lava field we were exploring.
I'm Professor Dave Cullen from Cranfield University in the UK. I started off kind of from my PhD onwards involved in biosensor bioanalytical technology, initially in the field of applications to medical and environmental applications, and then of relevance to this particular campaign about 10 years ago. I kind of stumbled across what's now colleagues in the space planetary exploration sector, and we started thinking about the possibility of using biosensor bioanalytical technology, not for medical and environmental applications, but in planetary exploration, trying to detect life in extreme environments. The initial context was detecting life on Mars.
My name is Thomas Cullen. I go to school in the UK, and my dad is Professor David Cullen.
My name is Malene, and I'm studying biology at the University of Copenhagen, where right now I'm finishing up my master thesis on virus-host interactions in archaea and archaeal viruses.
My name is Eddie Suderman. I'm an astronomy and astrobiology PhD student at the University of Washington in Seattle. My research involves looking at Earth as an exoplanet and modeling terrestrial planet atmospheres to better characterize terrestrial planet spectra when we actually get observations from space-based telescopes.
My name is Elena Amador. I'm from the University of Washington in the Earth and Space Sciences Department and the Astrobiology program.
So I'm Chang Yin. Now I'm working my postdocs at the Stockholm University. So I'm molecular biologist. My work is focused on the ribonucleotide reductase. It's a very important enzyme for the very beginning of life.
I'm Amanda Stockton. I'm a NASA postdoctoral program fellow at the Jet Propulsion Laboratories in Pasadena, California, and I'm developing miniaturized microfluidic instruments to look for signs of organic chemical signs of life on other planets.
So my name is Morgan Cable. I am a technologist at the NASA Jet Propulsion Laboratory in Pasadena, California, and I'm a chemist by training, got my PhD at Caltech, and I'm interested in how to detect different organic molecules, whether that is in situ using microfluidic techniques or other ultrasensitive techniques, or if we want to do remote observations to look for signs of life or signs of organics from orbit.
I'm Adam. I'm a PhD student at the Open University in the UK. I'm working on the interaction of the atmosphere and subsurface of Mars and working on the ExoMars Trace Gas Orbiter.
My name is Diana Gentry. I'm a graduate student in mechanical engineering at Stanford University, working in a lab at NASA Ames Research Center. Part of the research that I do is device design for biological sampling in extreme environments.
I'm Gayatri Murugesan from the University of Turku in Finland. I did my PhD there, studying on biofuel production from cyanobacteria from the local environment in Finland.
Our project kind of has a few different aims. I think we're trying out some new analyses techniques that could potentially be used on a rover or by humans for human exploration of terrestrial planets. And then there's this really interesting component of remotely mimicking a Mars mission to a field site. And so we're trying to essentially just test out different methods.
First of all, we're trying to demonstrate that we can take the three techniques and run them not in a traditional university or institutional laboratory, but run them at a field site. So we're local to the actual sites we collect samples from. And of course, in this campaign, that's done here in a local school, about an hour's drive from the field sites we're interested in.
Normally, fieldworks are like that. You do a lot of sampling during some time. You take the samples back, and then you analyze them at home, and then you scratch your head and say, okay, we should have done maybe that. We should have done maybe this in addition, or we should not really have done that. And having a field lab in the area allows us to get a constant stream of data in, which then alleviates our decisions where to sample next and what problems to tackle next.
So the key thing about having the techniques here in the field site is that we can have multiple cycles of sample collection, analysis, interpretation of the results, and then go back and do further cycles, such that hopefully we have a much more efficient return from the field work than we would do via the traditional approach, which is still commonplace in scientific circles, which is to go onto a field campaign, collect samples, package them up, ship them back to an institutional laboratory, and then weeks or months later do the analysis, and only then find out whether potentially you've collected the wrong samples, there's been a problem with the preservation of the samples, or if there is any interesting results, having to wait until the next field campaign before you can then test them further.
We're going to divide into 2 teams, and then some people are going to go take samples every day, and the rest of the group will stay back and analyze the samples. And we've brought with us a field lab where we can do kind of simple techniques that are possible to do in the field or in a field lab, where we can get some initial insight into what we're dealing with. So that's, for instance, why we're doing the qPCR, so we can see what types of metabolisms are present, for instance, or we can see like the ratio of bacteria to archaea. We'll see more archaea than we'll see bacteria and stuff.
The rest of the week we'll go to different field sites in Iceland that we've kind of determined are of interest to us biologically and geologically.
In terms of the science questions that we're trying to answer here, we are looking at the effectiveness of a couple of different life detection and quantification techniques, and how sensitive, both how good they are at picking up differences picking up differences in similar looking types of substrates that nonetheless may have biological differences, and also what kind of heterogeneity you see on different spatial scales in the field.
What can we learn immediately in the field versus more in-depth analysis back in the lab? And this is kind of will inform our ability to say what a future Mars rover equipped with some of the techniques we're using can actually determine without actually bringing back a sample to Earth.
So I think a really great goal to have by the end of this expedition would be to have a working method for how, if we sent humans to Mars, how we would go about sampling a field site there.
To me, the goal of this expedition is to be able to inform future planetary missions, whether those are in situ missions where we're landing somewhere and we want to help decide if this place is the right place to sample, or if the place that we're sampling is good representation of kind of that whole area, or also to better inform remote observations. So if we're in orbit around some place and we take an image of an area, what can we learn from that? What can we tell from that image that then we can apply to things going on the surface?
The majority of the people who are here are kind of early stage career scientists, so either research students or kind of early stage postdocs. And so the other objective of this is to give this group, some of which have not been involved in such field campaigns before, experience of both the techniques and running field campaigns, so it helps them develop their own individual careers.